let's make a start. So, it gives me great pleasure to introduce this week's Zangwill Club speaker, who is our new assistant professor in the department, Varun Warrier. And I'm going to tell you just in a few minutes his journey of how he's got here. So in 2011 he got his BA in zoology at Madras Christian College in India. And he also, in the same year, got a diploma in biology. He then moved to London and in 2012 got an MSc at UCL. And he focused his thesis on neuronal ceroid lipofuscinosis, which you all know what that is, a form of dementia. But then he moved to Cambridge to do his PhD. And I had the privilege of supervising his PhD. So in 2014, he got his MPhil in 2014, taking a candidate gene approach to understanding the genetics of autism. But in 2018, he got his PhD, taking a different approach, which was genome-wide association studies. And actually, he sort of lived through a revolution in the field of genetics as we moved from candidate gene studies. Varun published a very important paper at that stage in the history of psychiatric genetics, basically debunking candidate gene studies as not reproducible. We don't have to throw out all of their prior knowledge, but certainly we've moved into a new era, which you'll hear about, where we have much bigger data, which we hope will be more reproducible. Along the way, he's held after his PhD, held postdocs and fellowships at St. Cat's, at the Autism Research Centre, at the Cellular Centre for Human Genetics. He's won many awards. The INSAR, International Society for Autism Research, Distinguished Dissertation Award in 2021. Slightly from the World Congress of Psychiatric Genetics, the Early Investigator Award in 2016. Even this year, he got the Gershon Pope of the Year Award from the International Society of Psychiatric Genetics. So you can see that this is a rising star. He's published over 40 papers, and this year he had a paper in Nature Genetics. So without further ado, Varun, if you'd like to get your first slide up, because we're recording this, so you can see the title of your talk. There we are. The genetics of cortical phenotypes, insights into organisation and development.
Thank you, Simon, for the very nice introduction. Thank you, Warren. Nice to see some familiar faces, and thank you for the very warm welcome so far. Just in case you're wondering, that isn't me. That's Richard, who's sitting right at the back. I just want to sort of reinforce that this was a very collaborative work between Richard and me. He's the neuroimaging guy, as you can clearly see, standing in front of the scanner. I'm not. I'm a geneticist. So if you have any neuroimaging questions, please ask him. Genetics also, you can ask him. So I don't have too many slides, but this is a genetics talk, so I'm going to sort of try and unpack it for you. And if you have any questions that can help you understand the slides, please ask right then and there. But otherwise, more sort of interesting questions and deeper questions, please leave it towards the end of the talk. So before the pandemic, when the world was new, Richard and I were sharing an office, and we got talking, and we thought, here's a really interesting idea, which is what exactly are these MRI measures really measuring from a biological standpoint. So Richard was looking at MRIs, I was looking at genetics, and how can we sort of combine both our expertise to sort of try and understand how we sort of map what you find on the scanner to what's the biological processes deep in the cells. And we enabled that by using large-scale data sets from multiple different cohorts, but particularly the UK Biobank, which many of you are familiar with. So let me see if this will work. Maybe not. So Let me sort of set the scene. What did we know before we started the study? We know that cortical structural phenotypes, so MRI structural phenotypes, are by and large heritable. So when we say heritable, it means that a proportion of the variance in the phenotypes is attributable to your genes. We know that there have been some studies investigating the common variant genetics, so the genetic contributions from common genetic variants of things like surface area, volume, cortical thickness, white matter tracts, and to a lesser extent functional brain networks. But there were, and this largely came from a pioneering collaborative effort called enrichment, which was established in 2008 when GWAS was still sort of in its infancy. But this has subsequently, because of the availability of large-scale data sets like the UK Biobank, researchers like Richard and me have been able to use this data set to sort of address some of these questions and not go through sort of these meta-analyzing loads and loads of small-scale data sets, which comes with its own inherent problems. But there were some sort of gaps that remain. Increasingly, neuroimaging studies have been looking at other cortical phenotypes, such as microstructural metrics and measures of foldings, and these have not really been investigated from a genetic point of view. So we wanted to ask what the genetics of these phenotypes are and how they relate to the genetics of other well-established cortical macrostructural phenotypes like volume and surface area. And more importantly, what are the underlying cellular and developmental mechanisms that give rise to it? So we know that brain development arises through highly choreographed, stereotyped developmental processes through multiple different cell types, but we don't know how this sort of complex stereotype processes ultimately lead to the phenotypes that you measure in an MRI scanner. And from a human evolutionary perspective, because again we know that brain development is under critical evolutionary selection pressure, what are the genes that contribute to the variance in these phenotypes? Is it all genes, or are there specific sets of genes which contribute to the variance in these phenotypes? And are these genes then also linked to any well-known clinical conditions? For example, do the same genes that contribute to variation, normative variation in surface area or cortical thickness, do they also contribute to something like macrocephaly or microcephaly? And finally, what can genetics tell us about the regional organization of these phenotypes? So we set about trying to address these questions. And let me begin, this is a schematic diagram. Let me sort of introduce you to 13 different phenotypes that we looked at. So using data from two cohorts with a total sample size of 36,000 individuals, the two cohorts were the UK Biobank, which is an older cohort of people predominantly between their 60s and their 80s, and ABCD, a much younger cohort of people, young children aged between 9 and 10. In both these sort of cohort, we had genetic data, and we also had MRI data, and using the MRI data, we measured 13 different global cortical phenotypes, and these are standard phenotypes such as cortical thickness, volume, surface area. Additionally, we also measure different measures of curvature and folding. So we had something called local calcification index, which is how folded the inner surface of the cortex is compared to an outer straight line. So that's a ratio. Mean curvature, Gaussian curvature, intrinsic curvature index, and folding index. So these are all sort of measures of curvature. And then using sort of TTI data, we also looked at things like fractional anisotropy, mean diffusivity, and using three Nordic metrics, so intracortical volume fraction, isotropic volume fraction, and orientation diffusion index. So some of these, such as mean diffusivity and ISOVF, are measures of water diffusion, so how easy is it for fluids to sort of diffuse within brain tracts, and the others are sort of measures of neurite orientation and density within the brain. We measured these phenotypes both globally and across 180 regions using a postulation scheme known as the Clauser postulation scheme. And we restricted our studies to individuals of predominantly European genetic ancestries for multiple reasons, but primarily because of technical limitations that we have. And then we sort of corrected for the total number of independent phenotypes using a matrix decomposition approach and defined an experiment-wide significant threshold. So using all of these data, we first conducted genome-wide association studies, so 13 genome-wide association studies for the global phenotypes. and then 180 times 13, which I don't know the maths for, but I'm sure one of you can figure it out, but 2,000 something regional phenotypes for all these different values at a regional level using the postulation scheme. And then using this, we sort of ask few other questions. Predominantly, the first question being, what's the inherent latent structure from a genetic perspective between these different phenotypes? How are they linked to development and cell types? What is the impact on genetic constraint and neurodevelopment? And what can we talk about, what can we identify from regional genetic topography using genome-wide association studies? So let's start with the first bit, which is the genetics of the global phenotypes. So as I mentioned earlier, we conducted 13 different genome-wide association studies of the global phenotypes. And we conducted first in the UK Biobank, so about 32,000 individuals. And in the UK Biobank, we identified 314 genome-wide significant loci. So these are loci that are statistically associated with the phenotype of interest. So phenotype is the MRI measure that we are measuring over here. And then we want to see how robust and reliable these findings are. So we try to replicate these findings in a second cohort, which is ABCD, which is a smaller cohort of children aged between 8 and 9 and 10. And what you can see over here, so over here I have the effect size of the significant finding in the UK Biobank, and over here I have the effect size of the same variant in ABCD. And what you can see is that by and large you find concordant similar direction in effect size between UK Biobank and ABCD, which is interesting because we know that genetic influences on brain structure changes over age, and we also know that brain structure also changes with age. This is at the level of the significant genetic variants. We can also ask this question across all genetic variants measured using something known as genetic correlation. So genetic correlation is very much similar to regular correlation, but you're not correlating your phenotype or the metric itself, but you're correlating the genetic signal. And what you can see over here is that the genetic correlation for all of these different phenotypes is significant and positive, so that's 0 over here. For some of them it's not very high, so things like mean diffusivity and isotropic volume fraction is not very high, and we know that these water diffusion phenotypes change continuously with age. So all in all, this sort of gave us confidence that what we found in the UK Biobank is largely generalizable in a slightly different cohort at a different age range. So given this, we then proceeded to meta-analyze the signals obtained from UK Biobank and ABCD. And over here is a common way to sort of plot this. This is called a Manhattan plot because it's meant to resemble the Manhattan skyline if you're lucky. Usually it resembles the skyline of Cambridge when you're not really lucky. But the way to sort of interpret this is as follows. So each dot on this represents a genetic variant, a single nucleotide polymorphism, and it's arranged on the x-axis based on position. So you have chromosomes over here, so 1, 2, 3, 4, 5, so on and so forth. And on the y-axis is the statistical significance, so the p-value, negative log 10 p-value. So the higher up it is on the skyline, the more statistically significant it is, and the lower down it is, it's not statistically significant. And we can define two thresholds of statistical significance. So the dotted green line is called a genome-wide significant threshold. So that's p less than 5 into 10 power minus 8. And because we were running so many different tests, we additionally corrected for the number of tests, and we had a more stringent statistical threshold of significance, which is the red line over here, which was 5, p less than 5 to the power of 10 to the power of minus 11. So what you can see over here is that when we use both of these statistical thresholds, we identified 360 something genome-wide loci across all these 13 phenotypes, and 89 of these were significant above the red line, so at an experiment-wide significant threshold. 17 of these loci were novel, and I put novel in quotation marks because what is novel keeps changing. And we sort of looked these SNPs up, these sort of genetic variants up in multiple different databases, and we couldn't find any association with any neuroimaging phenotype in those 4 databases. So having done this and having sort of established that there is indeed a genetic signal over here, and that these phenotypes are genetically meaningful, we then turn to our next question, which is what can the genetics tell us about the shared genetic relationships between these phenotypes? Do they all emerge from a similar underlying genetic profile, or are they all genetically different? So how are these 13 global phenotypes genetically related to each other? So our first port of call to try and address this was a simple correlation heat map. So you have a heat map over here. On the upper triangle, you have the genetic correlations, so how similar they are genetically. And on the lower triangle, you have the phenotypic correlation. So that's just regular sort of correlations. And on the diagonal, you have the SNP heritability, so the proportion of variants in the phenotype that's explained by all the SNPs. We have clustered this particular heat map based on the genetic correlation. And if you sort of look at the clustering over here, you can reasonably easily see that there are few distinct clusters. So there's this one large cluster over here in the bottom corner, which involves phenotypes related to cortical expansion. So things like volume, surface area, few measures of folding and curvature, so local gyrification index, intrinsic curvature index, folding index, so on and so forth. And just above this, there's a smaller clustering of two measures related to curvature. So we have mean curvature and Gaussian curvature. And then we have the two water diffusion phenotypes that tend to cluster together, mean diffusivity, ISOVF. And finally, there's a fairly loose cluster over here of cortical thickness, fractional anisotropy, ICVF, and ODI. So these are all measures to do with neurite density and orientation. Now, one of the interesting things that you can see from this particular heat map is that with the exception of cortical thickness, almost all the other phenotypes tend to have a spiky correlation profile. So they sort of correlate with some phenotypes, but they have very limited correlations with other phenotypes. The exception is cortical thickness, which seems to have moderate correlations with almost all phenotypes. This sort of makes sense if you sort of think of cortical thickness emerging from multiple different cell types. It's literally a layering of multiple cells on top of each other, which gives rise to cortical thickness. So we wanted to sort of try and formalize this relationship a bit more and ask what indeed is the latent structure amongst all these phenotypes. And we proceeded to sort of do this using a toolbox in the psychologist's arsenal, which is structural equation modeling. I'm sure you're all familiar with structural equation modeling, but we use a modified version of this called genomic structural equation modeling, which we sort of modified, we didn't modify, someone else did. But we use that to sort of look at the shared latent profile based on shared genetics. We had to exclude cortical thickness for multiple reasons, including the fact that it didn't really cluster well with any of the conditions. But after excluding cortical thickness, what you can see is that there are 4 different latent phenotypes over here. So the first one, as you would have guessed, pertains to cortical expansion. So folding index, intrinsic curvature index, surface area, LGI, volume. There's a second latent trait that sort of pertains to curvature. There's a third latent trait that pertains to neurite density and orientation, so ODI, ICVF, fractional anisotropy. And finally, a fourth latent trait that pertains to mean diffusivity, water diffusion, so mean diffusivity and ISOVF. And all of these latent traits are moderately correlated with each other, not substantially, but moderately. And what this sort of tells us is that we can go from these 13 different cortical phenotypes, and then we can essentially distill them to 4 or 5 underlying latent dimensions that capture bulk of the variance in these latent phenotypes. We can also ask this question from the point of view of only the significant loci. So if you look at the significant genome-wide significant hits and ask to what extent they share between these 13 phenotypes, we can create a map like this, which is called an upset plot. So we use the Bayesian algorithm to look at the extent of sharing over here. And what you can see is that once you restrict to the genome-wide significant loci, there are quite a lot of loci, so 13 of them to be precise, which are shared between the cortical expansion phenotypes, so LGI, volume, surface area, so on and so forth. There are 6 of them that are shared between the water diffusion phenotypes. There are 2 of them that are shared between fractional anisotropy and ODI. So you can see that what you see at a genome-wide level is reflected when you just look at the experiment-wide significant SNPs, again sort of giving us confidence in what we have found. So the shared genetics between these different phenotypes can represent multiple different things. One potential hypothesis is that there's an underlying causal mechanism upstream which sort of causes your cells to sort of divide in a certain way, which ultimately leads to these phenotypes that you're seeing. The other thing is these genetic variants independently contribute to these different phenotypes. So there's a causal relationship and there's independent shared genetics that doesn't represent the causal relationship between these phenotypes. So we want to dive a bit more into that, and in particular, given this particular cluster of these cortical expansion phenotypes, we wanted to ask if there was any causal relationships between any of these phenotypes. In other words, the cellular determinants of one phenotype, for example, surface area, were they causal for any other phenotype, for example, lateral clarification index. And to test this, we turned to what we knew about these phenotypes at a cellular level, especially with regards to folding. So we knew that the question of how folding arose in the brain was a long-standing question. There was quite a lot of interest in it, and there were multiple different theories which try to explain folding in the brain. One particular cellular explanation for this is something called differential tangential expansion. So it's a cellular theory of cortical folding. So pretty much when the brain develops, at the earliest stages of brain development, there are these cells called neural progenitor cells, which are pluripotent. So they can give rise to any different types of cells and give rise to almost all neurons, all the excitatory neurons, and most of the oligodendrocytes that you find in the cells. So we know that these sort of differentiate into different types of progenitor cells. These include a specific type of cells called radial glia, so that's over here, and a specific other type of cells called intermediate progenitor cells, or IPCs. Now, within radial glia, there's again a very specific type of radial glia which are expanded in humans, and this is subtype that's called the basal radial glia or outer radial glia, that is in light blue over here. And these are highly pluripotent. They engage in multiple different cell cycles of cell division, ultimately leading to multiple different cell types in humans. We know from previous cellular work that these basal radial glial cells are differentially distributed in the cortex. There are, in particular with relation to force, as you can see in this figure over here, there are more radial glias under thyroid than under sulci. So you can see greater distribution over here than over here. So one popular theory of folding is that the differential distribution of these basal radial glia in the developing cortex means that different parts of the cortex has got different proliferative capacity, and subsequently there are different number of cells that sort of arise. And we can then test that using a method known as Mendelian randomization. We know that these different radial glia ultimately give rise to surface area because their ultimate purpose is to increase the surface area of the brain. So that way, if you look at the genetic determinants of surface area, we could ask if the genetic determinants of surface area is causal for things like folding measures. We use a tool in the genetics toolbox called Mendelian randomization, which tries to mimic randomized control trials using genetic instruments. And I'm going to sort of try and walk you through these graphs. So essentially over here we have the effect of the genetic variant on surface area, and over here we have the effect of the genetic variant on something else, so in this particular case folding index, and over here intrinsic curvature index, and over here local clarification index. And what you have over here is different ways to calculate the slopes, and what you can see from this particular graph is that with greater increasing effect of a genetic variant on surface area, it also increases the effect on folding index over here, which suggests, because we're looking at genetic variants, it suggests causality. It seems to suggest that genetic variants that ultimately lead to surface area are causally related to increasing folding index, local clarification index, and intrinsic curvature index. We can flip the question, we can flip the direction of causality and ask if the genetic variants that underlie folding index or intrinsic curvature index or local clarification index, if they are causally related to surface area. And in this particular case, you don't find really consistent results. So this line has a significant slope, but if you look at these two different methods, you don't really find significant slopes over there. So we ran a few other experiments to really try and robustly confirm these results. And overall what we can see is that genetic variance underlying surface area is causally related to measures of folding, which provides some evidence for differential tangential theory of cortical folding. So having looked at this, we then wanted to ask, what do these different profiles or these phenotypes, how do they emerge during development, and what are the underlying cell types that underlie these different phenotypes? So as I mentioned earlier, we know that brain development or cortical development is very stereotyped, choreographed set of processes. So we know around conception there is neurulation, and over here we have time in gestation weeks. And then around mid first trimester all the way to mid second trimester, we have neuronal proliferation. That's when you generate a lot of cells. These cells differentiate into their distinct cell types, so neurons and oligodendrocytes. And once they differentiate, they migrate to their respective locations, so that's neuronal migration. And once they migrate, they form synapses, so synapse formation. And given that there's excess of synapses, there is programmed cell death and synaptic pruning, which all continue postnatally as well. So a whole lot of early processes happen prenatally, and then there's processes that happen neonatally and postnatally as well. So we wanted to ask what these different processes, if you look at our GWAS signal, what do these GWAS signal capture in terms of these particular processes? And it's not an easy way to try and investigate this. But one way we can try and investigate this is to take all the significant genes that we have identified from each of these GWAS and ask during which period of brain development are they expressed. What can we say about the expression of these genes during brain development? And this is precisely what we did. So let me again walk you through these graphs. So as you can see on the y-axis or the x-axis we have conception weeks. So this is first trimester, second trimester, third trimester. This is around birth, and beyond this are years. So this is infancy, childhood, so on and so forth. And on the y-axis we have the average expression of the genes that are significantly associated with these phenotypes. So I have these 13 different phenotypes. Their mean expression across time as measured in the developing brain. And you can immediately see that there are two slightly different patterns over here. So if you look at the gray matter macrostructural phenotype, so that's the cortical expansion phenotypes and the curvature phenotypes, you find that the genes that are associated with these tend to express and peak around the mid-gestation period, somewhere over here. And then they tend to their expression tends to decline postnatally and continue to decline postnatally. Whereas if you look at the cortical gray matter microstructural phenotype, so the metrics, for example, they tend to peak just around birth, and their expression is more stable postnatally, and they don't really tend to dip all that very much compared to the macrostructural phenotypes. And this again ties in with what we know about many of these phenotypes. We know that process of myelination continues postnatally as well, and we think that this is driven by the differential pattern of gene expression over development. So that was during development, but what about the cell types that underlie these different phenotypes? So we can then ask if these GWAS signal are enriched for any specific cell types in either the developing brain or in the postnatal brain. So over here on the x-axis I have multiple different cell types in the developing brains. So these are all radial glial cells, so these are all progenitor cells. We have neurons over here, excitatory neurons, excitatory mature neurons, excitatory mature upper layer neurons, excitatory deep layer neurons, and we have inhibitory neurons over here, and then we have other cell types over here, so oligodendrocyte, precursor cells, endothelial cells, so on and so forth. So all of this is in the developing brain. And what you can see clearly from here is that if you focus on the cortical expansion phenotype, so surface area in orange and volume in light blue, you can see that these phenotypes are enriched in cells that are related to neuronal proliferation. So things like glial cells and progenitor cells are enriched for these signals. But if you look at the gray bar, which is related to cortical thickness, you find that cortical thickness phenotypes is enriched in the excitatory neuronal cell types. So you need to suggest that there are different cell types that underlie these different phenotypes. Thickness emerges from things like neurons, whereas surface area and volume emerges from early progenitor proliferation. We can ask the same question using cell types in the postnatal brain. So over here the cell types are much more simpler. So we have astrocytes, microglia, oligodendrocytes, and neurons. And what you can see is if you look at the cortical microstructural phenotypes, so for example things like intracellular volume fraction or isotropic volume fraction, you find that they are enriched for oligodendrocytes predominantly, but you don't really find an enrichment in any of the other cell types. Well, if you look at something like cortical thickness or mean curvature, for example, they're enriched in most of your cell types, like astrocytes, oligodendrocytes, and neurons. So again, in the postnatal brain, this seems to highlight slightly different cellular processes, where things related to cortical microstructure are enriched for oligodendrocytes, which we know are related to myelin, whereas things like cortical thickness and mean curvature emerges from multiple different classes of cell types. So having looked at cell types, we then turn to our next question, which is what are the genes that sort of underlie these different phenotypes? We know that these are very complex phenomena, and we sort of hypothesize that if there are any genes that are linked to these phenotypes, if they are disrupted, then they are likely to lead to severe, profound neurodevelopmental conditions. Right? So to look at this, we first ask if there are any signatures of constraint, if these genes that are linked to these phenotypes are under evolutionary selection pressure. Are they under positive selection or are they under negative selection? So negative selection would mean that if there are any new mutations that arise in these genes, then these new mutations will be deleterious and these variants will be removed from the gene pool over here. So on the y-axis over here we have selection, so anything below 0 is negative selection and anything above 0 is positive selection. And over here I have the mean selection and the standard errors associated with the mean selection for these phenotypes. And what you can see is that most of the phenotypes are under negative selection, so that means they do not encourage any new mutations arising. And this is particularly true for the cortical expansion phenotype, so things like cortical thickness, surface area, folding index, etc. are under fairly strong negative selection. But if you look at the cortical microstructural phenotypes, you don't really find any major selection or significant selection, which seems to suggest that microstructural phenotypes aren't under major selection pressures, whereas macrostructural phenotypes are indeed under selection pressure. We can also ask this slightly differently. We know that in the genome, in the human genome, there are about 20,000 or so genes. We know that 2,000 of these genes are highly constrained. In other words, you find fewer mutations than you would expect by chance. So these genes are called constrained genes. If you have a mutation in these genes, the odds are that the person carrying this mutation has got a severe neurodevelopmental condition, or that the mutation is deleterious fetally, so it leads to sort of spontaneous abortions, terminations. So we looked at the enrichment of these different phenotypes to see if the genetic signals were enriched in any constrained genes. And yet again what you see is that genetic variants associated with cortical expansion phenotypes like surface area, volume, so on and so forth, were significantly enriched in constrained genes. And we can restrict the constrained genes to about 280 or so genes linked to neurodevelopmental conditions, so severe developmental disorders, including intellectual disability. And again what you see over here is that cortical expansion phenotypes, volume and surface area, were also enriched in genes that were linked to severe neurodevelopmental conditions. So altogether it's sort of telling a similar story in that cortical expansion phenotypes, which arises from early mechanisms during brain development, the genes that underlie are constrained, they're under strong negative selection pressure, and these genes are enriched for genes that are constrained in the human genome and linked to neurodevelopmental condition. But we wanted more direct proof. So one of the questions that we had was, do the genes that underlie non-native variation in brain size, so non-native variation in surface area and volume, are they linked to genes? Do they also contribute to non-native variation in brain size? So do they also contribute to microcephaly and macrocephaly, so the extreme ends of the brain size? And we tested this. We tested this using a method called polygenic scores in two different cohorts. So this is DDD, a cohort of children and adults with profound neurodevelopmental disabilities. And a second cohort called SPARK, which is a group of autistic children and their siblings. And we generated polygenic scores, so a genetic likelihood for volume, surface area, and cortical thickness, and we regressed them against macrocephaly and microcephaly. So over here on the x-axis, I have the odds ratio and the confidence intervals associated with them. So what you can see is that increasing polygenic scores for volume and surface area are associated with macrocephaly, whereas decreasing polygenic scores for volume and surface area are associated with microcephaly. Whereas for cortical thickness, which is not a cortical expansion phenotype, you don't really find a significant association. So it was a negative control, and we were pleased to find that this negative control was showing consistent results. We took it a step further. So in the DDD cohort, they had measured head circumference among the kids, and the fraction of the children in the DDD cohort, about a third of them, had a genetic diagnosis. So you knew what the genetic cause of the severe developmental disorder was. And we asked in the kids in the DDD cohort who had a genetic diagnosis, so that's in red, and who had no genetic diagnosis, if polygenic scores for surface area and volume were associated with head circumference. And indeed what you find is that regardless of whether you had a genetic diagnosis or not, polygenic scores for surface area and volume were associated with head circumference. So increasing polygenic scores for surface area and volume increased your head circumference. And this was quite interesting because up until this point, the hypothesis was that macrocephaly and microcephaly were caused by monogenic forms of conditions, so single gene mutations in these very, very rare genes. And over here what we're sort of showing is that it's not just single gene mutations, but it's a polygenic contribution. So a lot of different genes contribute in small amounts towards macrocephaly and microcephaly as well. And finally, a final bit of the research investigation was to try and make sense of the genetics of regional phenotypes. So all along I was talking about the 13 global phenotypes, but we also had 180 to 30 regional phenotypes, and we had all this data, and we were like, what can we say about these things? How can we make sense of all of this data? So let me walk you through the headlines. So for the regional GWAS, we identified 4,263 experiment-wide significant loci. These loci are experiment significant signals, genetic signals. These genetic signals were modestly enriched for constrained regions of the genome. So you are more likely to find genetic signal or genetic hit in regions of the genome where you are less likely to find mutations in general. But this 4,263 loci clustered into 456 semi-independent regions. So if you sort of map them all onto the genome, they all sort of clustered into 456 regions of the genome. We did not correct for global phenotypes. I know that among the neuroimaging community there's a robust debate of whether you control for global phenotypes when you're looking at regional phenotypes, but we did not for a few genetic reasons predominantly. But we used a slightly different method called GWAS by subtraction, where you subtract the signal of the global phenotypes from the genetic signal for the regional phenotypes and then see what you find. And what you find is that 53% remained experiment-wide significant and 95% remained genome-wide significant, suggesting that most of these loci have regional effects and they're not just driven by global effects. But we can ask, given that there are regional effects, we can ask if these regional effects cluster on the brain. So we identified approximately, so one of the questions is, let's sort of look at this particular graph over here. So over here you have 2D images of the brain. And we wanted to ask if in a particular significant locus, if it was associated with brain regions randomly in the brain, or if they were associated in any sort of topological manner. So brain regions that are closer together would have a significance, the same significant variant. And this particular graph, the variant over here that was significant was this particular variant for folding index. And what you can see over here is that regions in the dorsal part of the brain were significantly associated with this variant for folding index, but not necessarily for the ventral part of the brain. And similarly for intrinsic curvature index also, you find significant effects in the dorsal part of the brain, but not really in the ventral part of the brain. So I'm not really going to go into details of this because it'll take me a few more slides to sort of really explain. But the take-home message is that even among regional loci, there are distinct effects that are guided by topology. So regions that are closer together are much more likely to share a genetic variant, whereas regions that are farther apart from each other are less likely to share a genetic variant. And finally, given all of this, we wanted to look at regional organization of these different phenotypes. So we know that the brain is organized in different manner, but if you look at the shared genetics, for example, for surface area between these 180 different regions, what can the shared genetics of surface area among these 180 regions tell us? So we first created a genetic correlation matrix, region by region genetic correlation matrix, and we compared the genetic correlation matrix for these different 13 phenotypes using something known as cophonetic similarity. And what you can see over here, the cophonetic similarity based on genetics on the top upper triangle, and cophonetic similarity based on phenotypic information on the bottom triangle. And yet again over here you can find some sort of clustering happening. So you find that at the bottom there's a large cluster to do with curvature and folding metrics, so mean curvature, cortical thickness, folding, all tend to sort of cluster together. There's another cluster to do with cortical expansion phenotype, so volume, surface area, local clarification index. The water diffusion phenotypes tend to sort of cluster together, and then there's this sort of weight cluster which is not really clear with the sort of other sort of neurite density and orientation phenotypes, which suggests that genetically, in terms of regional organization, there are some distinct patterns that emerges which are shared between different sets of phenotypes. We tried to make sense of what these patterns were by extracting the first principal component from the genetic correlation matrix and projecting them onto the 2D graphs of the brain. And let me sort of draw your attention to two particular ones, which is local clarification index. So if you look at local clarification index, you can clearly see a directional effect of the first principal component of genetics. But then if you look at isotropic volume fraction, again you can find a directional effect, but it's an entirely different direction. So this is inferior superior, whereas this is in an anterior posterior sort of a direction. So you find very different directional effects from a genetic perspective. And what's interesting is that we know that early cellular processes that drive brain formation are directional. We know that there's a clear directionality to early mid-gestational processes in the brain. And then around birth, there is no directionality in gene expression. It's fairly random in the brain. But then if you go post-adolescence and into adulthood, again you find a directionality, but in a slightly different direction. And this seems to be genetically encoded, as you can see from this particular graph. So yeah, so that was essentially some headline findings from our work. And I'd like to wrap up by thanking a lot of people. As you can see, it was a collaborative effort. We have a preprint available online should anyone require any further details. Thank you.
Thank you very much. It was a wonderful talk. I just need to check with the Cellular Club organisers. Will we record the Q&A afterwards?
Yes, it's been
okay. So we'll do that. So, terrific talk. What we normally do at this point is invite anyone who needs to leave at this point, because we then can stay for half an hour or so for questions and discussion. So those of you who need to leave, this is your moment to sit away quietly. The rest of us will see you soon. That's great. Whilst some people are leaving, can I invite anyone from the audience to ask questions? This question, yeah, go ahead.
This is a very basic question, but those Manhattan plots and GWAS, are those like, so you look at univariate relationships, right?
Yeah.
So just one, why didn't you look at the interaction effects between
I think great question. Just computational difficulties, right? So we're looking at approximately 50 million genetic variants, and then the moment you sort of scale up to sort of interactions, that's 15 million into something. I don't know what the something is, but I know that it's it's pretty exponentially high. Yeah.
Go ahead.
Great. Quick question about the section where you explore the relationship between the different phenotype-associated genetic signals and their expression through gestation.
Yeah.
Right. So the way that works, make sure I understand it right. So the way it works is that you've got gene expression data over that time window, different genes, and you link those genes to the results that you get from the GWAS. Is that how that works?
Slightly differently, where it's the other way around, where what we do is from the GWAS, we link the SNPs to genes to sort of identify what are the significant genes. And for those genes, we look at the gene expression data in the developing brain. So this is the brainspan atlas. And then we sort of calculate the mean expression of the significant genes in the brainspan.
Got it. So the brainspan atlas, how many genes does that have in it?
That has about 20,000 or so.
Because how many does your chip have on it?
So a chip would, because it's genome-wide, it would theoretically cover all 20,000, right? But the ones that are significant would be about, depends. Like, so in this particular case, I think like 400 for surface area, 80 for like fractional anisotropy or something like that.
I guess what I remember is that when you did the GWAS, was it 15 million?
SNPs.
Exactly. I see SNPs and genes, right?
Exactly.
Got it. So it's not a problem. Okay, I get it.
Yeah. So we need, the problem arises, and this is, now that you mentioned it, so you find two different graphs over here. So the challenge is, how do we map SNPs to genes? Because it's not straightforward, right? So the first way, so on the left, we have a method called magma, where you just use physical position and you sort of say, if a SNP lies anywhere between, X and Y of a gene, so 5 prime, 500 base pairs above the start and 500 base pairs beyond the end of the gene, then you map it to that gene. Whereas the second one is a slightly more sophisticated manner, where we know that the genome is not linear, it's not two-dimensional, it's sort of three-dimensional, and there's sort of contact points to it. And we know that 98% of the entire genome is non-genetic, so they don't lie in genes. So using the contact points, can we then sort of map the sort of non-genetic part of the genome to the genes, and then sort of use that information to sort of map genes. So that's a different method called H-magma. So we use both of them, and we sort of provide consistent results.
Got it. So in the end, it's not it. So I was wondering whether there's like an issue of granularity in terms of what you have available in the atlas versus what you have available from the GWAS. But it sounds like that's okay, because actually it sounds like those SNPs, but it's not really so many genes.
Exactly.
Thanks.
Other questions? John?
Just following that, do you have any large hits in the original GWAS that are so far from any gene that you can't identify them?
Yeah, quite a few actually. So the vast majority of them. So I didn't present it over here. So the challenge is, how do you go from SNPs to genes and identify genes? So and we used multiple different methods to sort of try and map that. So there were quite a lot which were intergenic, where we couldn't realistically map them to any gene. And then there were some where we had the opposite problem, where they were in gene-rich regions, and we couldn't sort of conclusively say which gene it was. Yeah, the vast majority. So to give you an idea, there were about 89 significant loci, and we identified about 50 or so genes with a degree of confidence. And of those, one particular locus on chromosome 17 tags a complex inversion region. So in that region alone, we identified something like 20 different genes, and we couldn't resolve that.
Okay. Other people who want to come in? Is that you've got another question
Oh, right.
Go ahead if no one else is wanting to ask them anything.
Okay, so when you've got 13 phenotypes, and that phenotypic, those phenotypic measures have a structure of their own right, so there's like a correlation structure between them. In lots of the results, it looks like the correlation structure for the genetics mirrors it.
Yeah.
Not perfectly, but really, really close, right? And part of me thinks like that's unsurprising because essentially what you're doing in the GWAS is you're exploring whether there are variances you can add up to explain that thing.
Yeah.
Then I was thinking, but what would the explained variance be between the genetics and the phenotype? And that even if the genetics are maybe explaining 5% of the variance, in a way that it maybe is more surprising that you get this really nice mirroring of the genotype and the phenotype. They both have the same kinds of correlation structures. Because if even like a good polygenic score, I guess, is explaining like 4 or 5% of the variance, then it's still quite compelling then when polygenic scores have the same structural relationships as the phenotypes that they're predicting. So is it always to be expected that whenever you have multiple phenotypes, whatever the relationship between the phenotypes you see will be mirrored in the genetic structure, or do those things ever diverge?
That's a great question, and the answer is in this case the hypothesis is that they would mirror, that you would find a correlation, and if it goes away from that, that's when you're like, oh, that's surprising really. So we went in to sort of expect this sort of correlation. So what you find phenotypically is what you should expect genetically as well. Over here is for, and there's some interesting examples of this. One particular example is between depression and BMI. So in Europeans, on average, depression is correlated with gain in weight, so increased BMI. Whereas in East Asian, depression is correlated with decrease in weight, so decrease of BMI. And if you look at this genetically, you find exactly that. So when you do a GWAS of depression and BMI in European individuals, you find a positive correlation between depression and BMI. But when you do that in East Asians, you find a negative correlation. So so what you should find phenotypically in general is what you should find genetically as well. There are few examples where it's not necessarily true, where there was a bit of an aha moment, but that's usually because the phenotype emerges from multiple slightly opposing genetic signals. A bit of an aha moment, at least for me personally, Simon was immensely surprised, was the positive correlation between genetic variants for autism and IQ, or educational attainment. And when it came out, people spent a lot of time, a couple of years, trying to sort of figure out what it really sort of meant. But the fundamental aspect is that autism, there are at least 2 separate types of genetic variants that can independently lead to autism. So there are these hard-hitting, high-impact, rare variants, which ultimately leads to autism in the presence of other massive co-occurring developmental disabilities, including intellectual disabilities. So these variants reduce IQ. But then there are common genetic variants which tends to lead to autism without any of those, and indeed this class of variants is also associated with increased scores in education and IQ.
I guess that's the case where the phenotype maybe is more complex.
Exactly.
But yeah.
Who's next? Anyone?
Surely that's the same with depression, right? I mean, is that also very complex?
Very complex, yeah. It just picks up, I think many of these things, it is interesting. Genetic correlations are, at some level they are unsurprising, very unsurprising, that you can look at a genetic correlation map, you're like, yeah, I knew this, what else is it sort of telling me? But it's also a nice sort of proof that your genetic techniques are working at some level, because if you find something that's quite different from what you would expect, then you probably need to go back. It's probably false positives, and you need to sort of go back to your GWAS signals.
Maybe I can jump in. Given that we're in the Department of Psychology, and we've heard a lot about imaging genetics today, the phenotype that you were talking about was the brain and the cortex particularly. Can you can you describe how you might take this work further to look at clinical phenotypes and behavioural phenotypes?
Great question. So, well, we try to sort of scratch the surface of this a bit with our macrocephaly, microcephaly sort of work. An obvious sort of question is how does it sort of link to mental health conditions or sort of behavioral sort of phenotypes? And the answer is it's not straightforward. So we run genetic correlations, and there's sort of very clear associations with things like cortical expansion phenotypes and things like educational attainment, and so measures of cognition really. There is some less compelling evidence linking microstructural phenotypes and white matter phenotypes in general with neurological conditions, especially neurodegenerative conditions. But really, I think in the field it's been quite disappointing because when people did imaging genetics, they thought it would be slam dunk to sort of try and finally sort of find a link between using genetics between neuroimaging and sort of clinical and medical conditions, and you don't really sort of see that. So, Richard and I have been discussing about this, and we think one, because the brain is an emergent phenotype itself, it's more than the sum of its parts. We have been sort of discussing about how do we sort of use the sort of data to sort of then try and rebuild some of the complexity. We're sort of taking it all down into pieces now. How do we sort of put it back to sort of rebuild some of the complexity and then try and link that complexity back to these sort of phenotypes, clinical phenotypes.
You mentioned that one of your cohorts was an autistic cohort. I'm just looking at this slide here where you've got prenatally, you've got synapse formation and sort of growth of the brain, and that one of the fears, as you know, about the autistic brain is early overgrowth. So is there an opportunity to understand the mechanisms behind that early overgrowth in a condition like autism?
I don't know if we can understand it specifically in autism, but I think yes, early overgrowth more generally. It's interesting in that, so if you sort of look at this particular slide, you can see that genes which are linked to neurodevelopmental conditions, which includes autism with other profound co-occurring difficulties, tends to be enriched in things like surface area and volume. And then if we take a slightly different approach and look at genes which are transcriptionally dysregulated in autistic brains, they are sort of enriched in gene networks that are fairly active during the mid-gestation period. So just about the time of neuronal differentiation and neuronal proliferation. So we have a reasonable idea that at least in some forms of autism or autism with sort of co-occurring developmental disabilities, but that is a period where there's both sort of neuronal proliferation and increased likelihood gene networks which are active in that period tends to sort of increase the likelihood for developing autism. I personally am not really clear of what exactly is the mechanism, but that's a place to sort of look, do a bit more deeper investigation to see what is really going on over there and how are these sort of subtle alterations leading to sort of major downstream changes in gene regulation, leading to both autism and brain overgrowth.
Anyone else? Kate?
Very tentatively, because wow, I've got a lot to learn from you, so welcome to the panel, and I look forward to many coffees with you. Very tentatively, so but what about variation within typical development? So variation in IQ, variation in educational success, I guess, is the only ones I can think about. But what, variation in object permanence, milestones, other developmental milestones, rather than pathology or... Yes, that's my question. Not very articulate. To what extent does your work speak to variation within
normal? Yes.
Or is it more about sort of detecting, really aberrant? Is it better suited to detecting and working out the relationship with aberrant signals, if you like, than it is to understanding?
Yeah, the answer is not at all. I think it can be suited for many things. So one of the things, so this is normative variation in brain size, so we excluded people on the extreme, so 5 million absolute deviations or standard deviations. And the example that I gave you was a clinical condition, and that's just because of, sort of goes against the grain of current, thinking at some level. But if you look at the genetic relationships between these variants, these genetic variants and variants linked to normative variation and cognition, we find fairly moderate genetic correlations, positive genetic correlations. So you're talking about 0.25 to 0.3 between cortical expansion phenotype, surface area, volume, and things like cognition. I am quite interested in trying to sort of link these progenic scores back to in a developing cohort, like say ABCD or TEDS or ALSPAC and things like that, and see how they sort of link to things like age of first words and these sort of developmental milestones. I suspect there might be a link over there, but parsing that link from, because IQ also has got, or general cognitive abilities also have a profound impact on those developmental milestones. Yeah. So it's going to be a bit of a complex statistical problem to sort of try and disentangle what's specific and what's sort of shared between general cognition and brain development.
Go ahead.
It's my last one, so I only got one last question.
That's what you said last one.
And this one might really just reveal real stupidity. So one of the challenges of studying development is that everything tends to kind of mush together. And so when you find, so let's say in ABCD, we find a relationship between certain genes and phenotypes, brain or cognition or whatever. And the temptation then is to conclude that those genes have some kind of causal influence on that phenotype, and that's why there's a relationship. Of course, the challenge is that their parents also share an awful lot of the genetic material. And how do you disentangle the notion that actually the reason those things are related isn't because the genes cause those things, it's because of the proximal environment that parents create. So things like books in the home, we always thought was a really important thing for early literacy. Let's get interventions, get more books in kids' homes, because it turns out that that's predicted by all sorts of polygenic scores for things like cognitive performance. So how do we go about disentangling what is genuine genetic causality versus what is it about people's biological parents who are generally not always but often related to the people they're caring for and the environment that they create, and that's why there's a parent-genetic relationship?
No, that's a great question, and there's a lot of interest in the field about relating to that really. So you can sort of, the sort of catchy term for this is counterintuitively genetic nurture, which is interesting. It's an oxymoron at some level, isn't it? Because nurture is meant to be fairly non-genetic at the superficial level. So you have statistical models where you can sort of parse this out. So you can, obviously the trick is to sort of study within families, right? So you sort of assume that siblings share certain environment, and you have now data where you can calculate genetic scores in both parents and also in the child. And you can use essentially combination of structural equation modelings and polygenic score to sort of pass it out. So there are, you can sort of think of it within a family, intrafamiliarly, two major dynamics. So there's the passive gene-environment correlation, where, as you rightly said, the books are there, then sort of my sort of nurturing environment is there, so smart, educated parents are more likely to sort of fill their house with sort of materials that can promote the education of the child and things like that. But there's also this sort of reactive or evoked gene-environment correlation, where, a child, parents might spot a real talent in a child and be like, okay, let's put the child for this class, or let's sort of nurture this child, in which case it's not just passive, but it's sort of reacting to something that they see in a child. And then there is, the direct effects, where it's the child's own sort of genetic variants which sort of link to your fundamental outcome. So all of this is just looking at genetics. And then, of course, there is environment. And then outside of all of this is stochasticity, which is just a random chance. So the sort of intrafamilial dynamics, the moment you have sort of, you can look at it from different sort of combinations of designs. So there's adoption designs, twin designs, and trio genotype designs and things like which, where you can get at to an extent. These are not very precise. You find slightly different estimations of this. So to give you an example, about 15% of the genetic variation in educational attainment can be explained by SNPs. So the SNP heritability is about 15%. But then if you sort of toss this out into direct effects, so directly from the child's own genetics, versus indirect genetic effects, about 2/3 of this is direct effects, so about something like 10%, or like 9%, and the remaining 1/3 is sort of indirect effects. But tossing that indirect effects into active and reactive genome, so passive and reactive, is a bit more sort of challenging, and the methods are still sort of being developed to try and understand that.
Anything else from anyone? Maybe I can have a last question before we go for a drink, which is that we had one slide where you were talking about evolution, and you were showing that most of the clusters of genes or SNPs were under negative selection pressure, but there were some that, I think just 2 clusters that were under positive selection pressure. Can you tell us a little bit about those 2? What makes them different, and why is it advantageous to carry those clusters?
So it's interesting. So if you ask if they're statistically significant, they aren't necessarily statistically significantly under positive selection pressure. And then there's a power issue as well. So if you look at the 2 sort of phenotypes, FA and ODR, were the least well-powered GWAS is out there. But I think nevertheless, what I can sort of conclude, despite those 2 sort of caveats, is that there's a differential selection pressure where there are these sort of macrostructural cortical expansion phenotypes that are under fairly robust negative selection pressure. And the specific type of negative selection pressure is called stabilizing selection, where you sort of don't want values that go far away from the mean. So you want the values to always converge towards the mean. And I think this is a function of when these phenotypes really emerge. So the cortical expansion phenotypes tend to sort of emerge during early developmental processes, which are tightly regulated. And I think there's sort of less plasticity and less sort of scope for things going a bit awry during those early developmental periods, because they tend to sort of lead on to several things. So there's a knock-on effect in development. Whereas, especially water diffusion phenotypes, they tend to sort of emerge and they continue to sort of develop postnatally well into your adulthood as well. And I think it's because these are slightly late emerging phenotypes, they're not under such significant negative selection pressure. And a really interesting example for this is if you look at the effect sizes of common genetic variants on average and minor allele frequency, we know that the more common genetic variants tend to have smaller effect size on genetic conditions. The major exception is the ApoE locus for Alzheimer's, and because that sort of plays a role postnatally after your reproductive period, it's not under fairly significant negative selection pressure.
I'm just going to pause to ask Richard, do you want to add anything as a major collaborator in this work?
Varun's covered pretty much everything. I was kind of hoping to get away with no question. No, I think maybe the only thing to add, and I think Varun's already spoken quite extensively about this, I think one of the very early findings that we had in this project was this sort of discrepancy between the microstructure of the traditional grey matter phenotypes and the white matter phenotypes, and this big difference in heritability between the two. Which probably also kind of speaks to the selection pressure element of it, that the idea that this grey matter structural backbone is in place very early on, whereas the sort of white matter microstructural features, they tend to keep developing for a really long period. So they're under the influence of many different factors, probably also environment, as Duncan was alluding to. I think that's something to sort of dive a little bit deeper into some of the future work.
Okay, thank you. And for those of you who don't know, Richard will also be joining the department as a new assistant professor in neuroinformatics. A very warm welcome to you too. So I think it just remains for me to thank you again, Varun, for a fantastic talk. I must just share a little anecdote, which is that during your PhDs and postdocs, we did a little experiment. We put Varun and Richard into the same office, and Varun had come speaking one language, which was genetics, and Richard had come speaking another language, which was MRI. And after about 2 years, we opened the office to see what had happened, and found that they both learned each other's languages, and the result, as we saw tonight, was imaging genetics. So, thank you again, Warren.
Thank you.